Hello, everyone. Hello. We're going to begin now. We have a very exciting session. We're going to deal with the CNS, the central nervous system, and it's my great pleasure. I'm surrounded by experts. There are two of us who are virtual, and we're just now at the point of a revolution in how we approach the relationship between industry and academia. It's enormous potential, but the brain is pretty complex. I work on the brain. I'm a professor at MIT, and I can tell you that one of the great insights that's come lately is that it's sort of a new realization that the brain is made up of many, many, many tiny little microcircuits, and they're highly specialized, shockingly so that a few neurons can control a behavior. It's really an amazing thing. So how are we going to target these little circuits in order to control a particular problem? And you know that in our field, Alzheimer's and schizophrenia, Parkinson's, mood disorders, and then beyond those, our own behavior can be just slightly off. Anxiety, depression. These are very difficult and complicated things to deal with. So how industry is doing this is quite remarkable, and the opportunities for getting together with academic people it's huge. We really are in a time of revolution, all the way from molecular biology to the new methods we have to deal with the brain, to new forms of getting materials into the brain or into the periphery. These are extraordinary times. So it is my pleasure, really, to introduce the panelists who are here and who are not here. And we're going to start with a few comments by each, and then we're going to have a lively discussion, right? Mm-hmm. So we're going to start with Kelsey Martin. Kelsey is a remarkable person who at once has been at very, very high levels of academic medicine as a dean, and now has gone to head a large part of a very large nonprofit organization directed toward autism, and that's the Simons Foundation. And she's just come, and we are thrilled that she's there. She has the way, the experience to talk about this relationship like few others, because she's been on both sides. We then want to go to two people who really are going after precision medicine and targeting specific pathways. One is Michel Pelle. Hello. You're virtual. Hello.
Hello.
I'm sorry not to meet you in person, and we're glad to have you, managing director at Boehringer Ingelheim, and a real pioneer in taking small circuits seriously, and he's a very exciting example to talk about. And then Dan Grossman is here, and also is headed that way, and he has great insights in our prospects for new kinds of therapy all the way to psychedelics, which is pretty wonderful. Jay Stamatis, hello. We weren't sure that you would be here or virtual, but Jay has told me that he wants to talk about new ideas he has for how to really drill down into the way to relate industry to academia for efficiency and success. And then Diana Gallagher. Diana is head of external innovation at Biogen, an incredibly talented and experienced person. And she has, I think, great insight in what we really have to do in this field. As it was said just before in the previous panel, there are a lot of hard details to cover. So let's start with Kelsey. Thank you very much.
Well, you're welcome. So such a pleasure to be here. Thank you, Anne. And I'm going to give you my perspective as the former dean of the UCLA Medical School for 6 years, and then more briefly about running the Simons Foundation Autism Research Initiative, which I've been doing for 6 weeks now. So for me, the opportunities, I actually come as a neuroscientist to being the dean of a medical school, and the opportunities to advance CNS therapeutics that are just enormous. And in particular, by putting together clinical neurosciences with the advances that Anne was talking about in basic neurosciences and just the transformative enabling technologies that have emerged in the last decade or two, really start to make this a possibility. And I wanted to just give you one example of a project that we undertook at UCLA, and it was aimed at, it was called the Depression Grand Challenge. It was a project that emerged from the basic neuroscientists at UCLA, where they really tried to think about what's an area where we can address something that has high social impact, where if we work collectively across the campus, we might be able to make some inroads. And the idea that depression is one of the major global burdens of disease, and in particular at a university where individuals ages 18 to 25 really suffer from depression, it seemed like it was our responsibility to do that. So we set out a very large genetic and phenotypic screening aimed at 100,000 individuals working through our hospital system. And I'm not going to go through it. What I want to do is I want to just tell you some of the issues that arose from that experience. And it really was, first, that incredible need for strong partnership with clinical hospitals and clinics to be able to enroll patients. And the need to supplement any research with clinical care. I mean, there's a moral responsibility that if you're doing screening for depression, you've got to be able to identify individuals who are at risk for suicide and intervene immediately. There were lots of questions about how does one focus on, is it better to focus on small populations? One part of the project was looking at postpartum depression, more narrowly focused, but then there was a real desire to have a much larger, more diverse population, and how do we manage that? And how do we address, and it sounded like some of these issues came up in the last conversation, issues of equity and diversity, and really engage the community. And in doing that, how do we address some of the stigma that exists around depression? So how do we use this as an opportunity to really talk about depression as part of the human condition, and how do we, and I have to say, since it's the Galien Forum, think about the French philosopher Georges Canguilhem about what's normal and what's pathological, right? So that's another really important issue for us. And then there were tons of really interesting scientific questions. This is obviously a polygenic disease, so how do we address this from really trying to understand or develop any treatments? How do we integrate clinical, phenotypic, and genomic data? I mean, that's a scientific challenge, and lots of opportunities for use of innovative technologies for phenotyping. And one project that was a great collaboration with Apple was to use really innovative technologies to monitor activity and sleep and voice, and to try to use that information. Another scientific question is, what's the utility of the preclinical models to something like depression? We use something called, forced helplessness. Is that really what depression is? And so, how does one model those sorts of, and these are some of the issues. And then I would say, finally, there's just an enormous contribution of social and environmental determinants of depression. Probably the largest is determinant of depression is childhood abuse. And so if we're really focused on the science and the biology of depression, we also have a moral imperative to not dismiss those others and to actually pay attention to our responsibilities there. I'll end by, my part about the academic component by saying that what I found really useful in terms of academic and industry relationships was working with our tech transfer office on developing an innovation fund where we got proposals and they had 2 tracks of review. One track was a scientific review by academics and the other track was a business review for looking at unmet needs and really teaching. And I think that was incredibly impactful. And I'll just end by saying that I moved, wonderful job being a dean, but I moved 6 weeks ago to the Simons Foundation. And the motivation for me there is that this is the largest private funder of research on autism, another very complex disorder that really teaches us about the social brain and about our who we are as humans, as social beings, and with the goal, the mission of really deepening the understanding, the diagnosis, the treatment of autism through funding really innovative, high-impact research. And it does so in 3 ways. One is by funding academic research. Another is by developing cohorts of families, children with autism, and that's so important because that's where you can really bring in that community voice, and you can really try to begin with the human experience of autism. And then the third is by creating resources that are useful for the research and the autism community. And so whether those resources include genomic databases, genetic databases, they include model systems, whether they're induced pluripotent stem cells, zebrafish, mice models, and trying to leverage those so that they're available to the entire academic community so that there's a motivation to be collaborative. So I think I'll end there. Thank you.
Thank you. That's a wonderful, wonderful, wonderful introduction. So, Michelle, where are you?
I'm here, and I'm trying to share a few slides.
Ah, okay.
Can you see my slide? Can you hear me? Yeah.
Yes.
Okay.
There.
Okay. The topic, first of all, it's a pleasure to be here, and thank to the Galien Foundation for the invitation. My topic will be a path to precision psychiatry, and we decided to enter the field of sorry. We decided to enter the field of psychiatric diseases around 2010, and it was a decision that against the stream, since considering the high failure rate during clinical development. So we decided to enter the field nevertheless, considering the remaining high unmet medical need, but also realizing that we need a novel approach in drug discovery enabled by a deeper understanding of the biology of the disease. To achieve this ambition, we decided to build on a new concept, which was developed by the National Institute of Mental Health, the Research Domain Criteria, or RDoC. With the RDoC concept, we are moving from the traditional DSM classification of disease. DSM stands for Diagnostic and Statistical Manual of Mental Disorders, which is based on the simultaneous occurrence of several, sometimes poorly related symptoms, like in depression or in schizophrenia, to a classification based on symptom domains like cognitive impairment or emotional dysregulation. And the drug discovery approach inspired by the RDoC concept is to investigate the link between specific symptom domains and specific brain circuits. And this is expected to yield more robust and testable therapeutic concepts. I will skip a few slides because I realize that we don't have so much time. I would go to the next. And as a first step, we've decided to focus our RDoc-based R&D efforts on the areas of motivation and emotion, cognition, impulsivity, and reward. And as previously mentioned, the RDoc-inspired drug discovery approach of linking specific symptom domains with specific brain circuits needs to be enabled by new translational technologies applicable in human and animals, and which need to develop and validate in close partnership between industry and academia. Though there is still much to develop in this field, it's interesting to see on this slide that many technologies can already be applied in both animals and humans, including wireless EEG, PET, fMRI, computerized cognitive testing, etc. Taking now the practical example of amygdala-driven emotional dysregulation. Emotional dysregulation is the consequence of an imbalance in the cortical limbic system, often driven by amygdala hyperactivity, as indicated on the right-hand side of the slide. This emotional dysregulation can be measured by monitoring the reaction of a test person to an external stressor like a fearful face. A normal person, for example, would react at the 30% picture between fear and sadness at the bottom of the slide. A person with a hyperreactive amygdala would show the response already to a 70% picture. What is interesting is that amygdala-driven emotional dysregulation is involved in the various DSM psychiatric conditions like major depressive disorders, borderline personality disorders, or post-traumatic stress disorders. This slide shows, the next slide, this slide shows how we identified patients with amygdala-driven emotional dysregulation across various DSM indications using an fMRI translational biomarker. We also see on the right of the slide, illustrated by the frequency of spikes, that TRPC4/5 cation channels increase the excitability in amygdala, and that the inhibition is expected to normalize emotional processing, and this is shown on the next slide. On this slide, you see how amygdala activation induced by an external stressor is attenuated by TRPC4/5 inhibitors in animals, and also how this translates into human. We have to admit that the external stressors and the biomarker used were different in these studies, but today, based on technology development, we would use very similar biomarkers in both animals and humans. In summary, I can say we've started a decade ago working with diverse partners to establish a novel drug discovery approach inspired by the RDoc concept and linking specific symptom domains with specific brain circuits. There is still much to do, though we see first approaches bearing fruits, and I've just introduced the approach that we co-developed with our partner Hydra Bioscience to normalize amygdala-driven emotional dysregulation with a TRPC4/5 inhibitor. We are now testing the TRPC4/5 concept in phase 2 clinical trials in major depressive disorders, borderline personality disorder, and post-traumatic stress disorder. I mentioned in a previous slide the role of new technologies. These technologies are applied to support target identification, patient selection biomarker, other translational biomarkers, including digital readouts, as well as a battery of tasks measuring in the clinic. This can only be achieved by working with partners from across academia and biotech companies across the whole value chain of our psychiatric R&D. A few of these partnerships are listed on this slide. I will only introduce 2 examples. The first is our collaboration with King's College London on cognitive deficits in depression and schizophrenia. And the second is the work of the PRISM Consortium as part of the Innovative Medicine Initiative, which is using smartphone monitoring of social functioning and parallel phenotyping with fMRI, EEG, and a range of cognitive tasks. And in doing that, they were able to identify, and we are part of it, of course, they were able to identify 3 new clusters that are independent of traditional diagnostic based on the DSM classification. In summary, we are convinced that RDoC enables a new approach in CNS research, which is closer to the biology of disease. We see this approach starting bearing fruits, though we fully realize that we still need to deepen our understanding of the relation between symptom domains and brain circuits by continuing to establish a network of collaborations across industry, biotech companies, and academia with a strong focus on translational science.
Thank you very much. Thank you so much. This is the future, and you all are real pioneers, and we celebrate this very, very much. There is another person, Dan Grossman, who is head of the Boston Consulting Group, and he's impressed me very much as having at once a realization of how doggone tough it is to work on CNS problems, but on the other hand, the enormous possibilities that already are almost in our hands. So Dan?
Thank you. Thanks for the kind words. I can't help being struck by the corporate senior executive who brought PowerPoint slides and the consultant who does not have any. It's quite an inversion. But if anybody wishes for any slides after, I can send you. I can send you many.
Now this panel is really getting good. Let's go.
So I mean, but I was struck as I heard about the topic for the panel in the first place. I was struck by looking back in time and thinking about if you tried to bring up this topic about fruitful progress in CNS disease, if we tried to do this 15 years ago in the mid-2000s, you could barely hold the panel, or it would be unbelievably depressing. And that's not to say that nothing was happening, but from the perspective of industry and industry output of new therapies, very, very little was happening. It was a very, very dark night in the field. And you saw that from the withdrawal or retrenchment of many of the large pharma companies, some very significant present company excluded, no question about that. But you saw, with a few exceptions, you saw companies either exiting CNS disease entirely from a research point of view, partnering out or licensing out what work in progress they had. In some cases, retrenching and at least shrinking what they were doing and focusing in a few very narrow areas. Particularly getting away from psychiatry entirely, saying, okay, well, maybe there's a few select things we can do in neurology, but we're moving out of psychiatry. And that would have been the story at that time. And now here we are in 2021, and it's very much coming out of this night into a real dawn where lots of exciting things are happening, some of which we just heard about. I'd like to just say a word or two about some of the things that we see that are happening that are so exciting and so promising, and a little bit about the role of either the actual or the potential role of industry and academic collaboration in making those happen. I won't go into great depth, but we'll be happy to take questions about some of them. I mean, obviously, we see a proliferation of new and exciting and potentially druggable targets, and it's that is the most traditional, most central role for these kinds of collaborations where academia has the capabilities, the types of resources, the talents, and the workforce to try to identify and then elucidate properties of new and interesting targets. And we've seen great drugs come from that. I'm particularly struck by the a proliferation of new therapies for migraine coming from anti-CGRPs and many more targets behind that coming for a disease that in those mid-2000s we would think, well, this is just something people are going to live with triptans, and maybe there was Botox at the time for that. You see a move now toward new discoveries of risk factors for the development of various neurological and some psychiatric disease. I'm struck. It's one of many points in perhaps to me the most notable broad collaboration between industry and academia, which is the Foundation of the National Health Institute's Accelerating Medicines program, which in its second round has enormously expanded the attention given to CNS disease, and in particular I'm very excited about what they're talking about for schizophrenia and risk factors for development of schizophrenia, which will, we would expect to improve patient care down the road. You see, a lot of progress in, of course, imaging, which plays a major role in understanding CNS disease and being able to accelerate and do higher quality clinical research. As new therapeutic candidates are being tested, it's hard to, you could never cover all of the different interesting and productive collaborations going on around imaging for CNS disease. Although I was excited to hear, I asked some of my colleagues about this, and I have a colleague who was part of a postdoc exchange program, not postdoc exchange, a shared postdoc program between Pfizer and Stanford, which yielded some, really exciting breakthroughs in longitudinal calcium imaging and visualization, which can be used for lots and lots of, for the study of lots of neurological disease. There's, I can't go through too many more of them. There's translational neurotoxicity biomarkers, where we see a variety of industry academic collaborations, a number of universities, a number of governments participating. Those are all, to me, somewhat, no question they're advancing science, no question that some of them will translate into improved patient care, but they are somewhat traditional. They're the enhancement and advancement of traditional life science research, traditional research tools, traditional approaches to identifying new therapeutics. There are two areas where they go beyond that don't fit the mold very well, and where I would say there's more uncertainty or more innovation required in the way that industry and academia collaborate. One of those, which I won't dwell on too much, is digital health, and specifically digital therapeutics, where if we say digital therapeutics could be making a big difference for patients in 2030, I don't think anyone has any question that it's going to be in neuroscience, possibly neurology, almost certainly psychiatry, where that will make a difference. And so far, that field has gone slower than we'd like. There's understandable skepticism about the effectiveness of some of these therapies. In some cases, it might be a self-fulfilling skepticism, which is a little bit concerning. But I think anyone working in the field would say maybe there aren't going to be digital therapeutics as any kind of standalone solution, but as something that enhances the clinical impact of the medicines being brought forward. I think anyone in the field would have optimism about that. But there's not a lot of clarity about where the relevant research into the workings of digital therapeutics is going to come from. There's a little bit in industry, but it's not the best of what industry is doing. There's quite a bit out in the tech startup community, but there are all kinds of problems with that translating into actual eventual clinically relevant innovation. And then there is a little bit happening in industry, but I think not nearly the amount that we would expect for a type of innovation that has so much promise for patients. There are deep scientific questions about how digital therapeutics for neurology, for psychiatry might work, why they might work, and trying to validate theories of why they might work, which are going to be needed before it makes any sense for industry to pour more serious resources into that space. So that's a concern where I see a little bit of a gap, although I'd love to be told by someone in the audience that I'm wrong about that, and maybe it is actually a much more thriving field than I thought. And then the last one that Professor Grable mentioned, and is by far the most exciting to me because I think it has the most revolutionary potential of any area of research in neuroscience over the next, say, 10 to 15 years, is the development of psychedelic medicine. And this is the use of compounds like psilocybin or LSD or ibogaine or MDMA that, in some cases, in many of those cases, were the objects of intensive academic and clinical research in the 1950s and the 1960s and into the early 1970s before it was basically shut down due to the drug war. It was sort of a side casualty of the drug war. And the results they were having then were promising. The field was suppressed for quite a number of years, and then in the early 2000s, it has started to come back in a notable way. And it's true, like in any field, there's a lot of hype behind it. There's a lot of money flowing into it, but real breakthroughs are on the way. And the one that is the one that's been most notable to me, and BCG has had some involvement in this, is that last year a company completed the first FDA registrational trial, a phase 3 study for the use of MDMA-assisted psychotherapy for post-traumatic stress disorder. And they had a population of subjects whose average time with continuous PTSD diagnosis was 15 years. Subjects coming out of the trial, the results coming out of the trial was that 60% of subjects in that study with that average of 15 years with diagnosis no longer qualified for a PTSD diagnosis at 6 months, and 68% no longer qualified for a diagnosis at 12 months. It's an unbelievable result that nobody in the psychiatric field who wasn't directly involved in that space would have expected. And now, we're expecting FDA approval for that MDMA-assisted psychotherapy in late 2023. Lots of challenges with bringing it to market for sure. But now you're seeing an explosion of additional studies of the various medicines that I named for, obviously major depressive disorder and some treatment-resistant depression. Beyond that, other somewhat intractable areas. So substance use disorder, ayahuasca for alcohol use disorder has been sort of practiced underground and outside the United States for a number of years. The ibogaine for opioid use disorder is being tested. There are very high expectations for eating disorders, lots of case reports and lots of investigator-initiated studies related to anorexia nervosa, bulimia, binge eating disorder, where there's, I hope I would not insult anyone if I said there is essentially no meaningful pharmacological treatment today. And then there are bolder and broader ideas to try to overcome some of the stigma both associated with psychedelic medicines and with psychiatric disorders. And I was excited to see yesterday an IRB approval to go forward with a study of MDMA in generalized anxiety disorder. And if that happens, it'll hugely broaden the field's appetite to further study the medicines. An interesting reflection here on the role of academia. Obviously, there will be, there are academic labs trying to understand why the medicines seem to work the way they do, how they can further enhance them, how they can, remove some of the undesirable effects of some of the medicines and enhance the efficacy even further. But there's another aspect to these where I think there's a role for academia, but less in the form of bench science and more in the form of academic medicine itself, innovation in clinical care. Because when I say that that phase 3 study that completed successfully and we expect to lead to FDA approval, it's MDMA-assisted psychotherapy. It is not write a prescription, give the patient the drug, check in with them regularly on how they're doing. It's an intensive course of cognitive behavioral therapy over a couple of month period in which the drug is used as a support. That's a very, very different paradigm for how care is given, and if it's going to be done well, and if it's going to be done consistently, and if in the real world we're going to replicate the results from the trials, then the community of psychiatrists and therapists is going to need to be given tools to understand what is the best way to actually conduct the therapy, not just what is the best molecule to give in what dose and in what frequency. And this feels like a role for academia that could be expanded quite a bit. Currently, that is being heavily studied in a few centers. I don't know if there's anyone here from Johns Hopkins who's involved in that center that's been stood up, or at NYU and Mount Sinai, they're exploring different psychiatric modalities to go with the medicines. Harvard MGH is doing the same, and then that's to say nothing of the international centers that are studying this. So I would have high hopes and expectations for increased interest, not just in the lab science part of academia, to bring to these collaborations, but also to the clinical science and methods of care.
Thank you, Dan. It's eye-opening to see all this come back and then just boom. It's very, very exciting. I can't take time to say we're working on that sort of thing, so I won't take that time. Jay, we're looking forward to you, even though you're virtual Vice President of ABFE, and as I said, you told me you have all kinds of new ideas you're going to tell us about. We're glad you made it. We weren't sure.
No, sorry, and thank you, Professor Graybill, for the introduction. And it's great to see all of you, and I'll meet all of you someday, hopefully. But thank you to the Foundation for the invitation to participate in the panel here. So look, first of all, I'm very humbled to be here. I mean, there's a lot of very esteemed researchers and people who have done amazing things in neuroscience and in their fields generally. So, I mean, look, I'm kind of honored in a way to sit kind of at the nexus of a lot of these elements here. I've got the ability and the responsibility to look at new science on one hand. That's what we're talking about a lot here, and the ability to bring resources from industry to kind of enable that science. And I think that's a fascinating place to be generally, and I think it's a really fascinating time to be doing this in neuroscience. At AbbVie in particular, we're involved historically for the past few years in neurology, and that's been an area of core focus for us. And over the past several years, we've built up a foundational neuroscience discovery unit in Cambridge, like in Boston, that's grown to more than 100 people from virtually none, maybe in 2016 or so. So this is an emerging area that's very, very important for us. And my view of it, from my seat, we've been trying to populate that research organization with new technologies and new ideas. Now, they're very much capable and currently producing many of their own ideas, but they have to come from the outside to get it jump started. And so, over the past maybe 5 or 6 years has been a really, really remarkable time in our industry overall, and in particular in neuroscience, but in our industry overall. I mean, they're just for perspective, in 2020, there's $16 billion in venture funding that's gone out to company formation, right? That is a huge amount of capital. And someone said to me in a meeting a few years ago, like, look, there's literally almost unlimited capital available for biotech right now. And that enables the funding and the run rate and the progression of ideas that maybe wouldn't have been funded before. So this is a very heady time for industry generally to take ideas that are being germinated in academia and bring them forward. And just for some additional perspective, since 2016, I was looking at this recently because I kind of had a sense this was the case, but just to put some numbers around it. Since 2016, there have been over 200 partnerships between industry and either academia or small companies, right? That's one of the leading areas across our industry. Oncology, I think, has more of it, but not many therapeutic areas would best that. And of those roughly 200 partnerships that have been put in place, half of them are preclinical or discovery collaborations. So that is industry resources, that's industry know-how, industry thinking that's sort of being leveraged into smaller companies or academic partnerships that can help them bridge this gap between discovery and the bench and how to industrialize, if you will. It maybe sounds like kind of not a nice word sometimes, but you do have to industrialize technology and medicine sometimes to bring it to patients, right? It's not gonna get to patients if it doesn't navigate the technical hurdles of manufacturing something or bringing it through the FDA, which is a whole skill set into itself. I mean, we heard about Boehringer's efforts to improve the probabilities and success rates of depression studies. Like, that is a very difficult place for industry to navigate historically, much, much lower probability of success in neuroscience or depression phase 3 studies than industry average. I think they're around 50-50 usually, compared to what I would usually say is 60 or 70% success rate for other therapeutic areas. So bringing that kind of innovation into the ecosystem is what industry, I think, can do sometimes. So, I mean, we have done this at AbbVie in lots of different ways. We do this through venture investing, many examples of investments we've made in startups and try to seed companies at very early rounds. The most common way for us to do this, and that I've been mostly a part of, have been in direct partnerships with companies that are in the early stages of their lives, where they have an asset. Like in the case of a company called Caraway that we partnered with this year, they have a TMEM175 inhibitor that will have utility in neurology, various neurology applications going forward. That's a very early program that they will take to a certain step, and then we will take it further after that. So a company called Mitokinin that has a PINK1 inhibitor, which we feel is a very important target, going forward. And look, our capital goes into that company. They will take it using their expertise to to the next inflection point for them, which I think would be an IND filing, and then we would take it from there using our industry capabilities. And another, I think, really interesting example of kind of this continuum from academia to company formation to biopharma industry. A new company that was formed last year called Capsida in Southern California. This was enabled by a venture investing group that we know, Westlake Village Biopartners in Southern California. This, I believe, technology came out of Caltech. This is a novel capsid technology, which in and of itself is innovative and important, but it needs a cargo. It needs targeting. And that's what we brought from AbbVie. So we have a broad collaboration across multiple targets to bring our cargo to use their capsids to get these therapeutics and these targets into the brain, right? That I think is one of the big frontiers that we have been trying to cross, literally and figuratively, to get therapeutics into the brain. And so, you know, this is a technology out of Caltech into a startup with venture financing, and now in a collaboration with the Discovery Center that I mentioned to you earlier for us in Boston, leveraging whatever capabilities we can bring and experience we can bring and resources we can bring to bring those across the line through the clinical trial process and to patients. So it's kind of a good example, I think, of how the whole model can work. And like I said at the beginning, I think this is a really, really remarkable time in industry generally, but for neuroscience more specifically, to enable science and bring all of these things together. And I don't want to end without mentioning some important collaborations that can be done at the basic science level, at the academic, with investigators in academia, because that I think is also a very, very important area too. I won't describe all the specific ways that we do that, but they're common, I think, to many others in the industry, and enabling good science with good funding and resources without strings, right? We're talking about sort of how can we sort of help bring science forward and bring some perspective to help science advance. And we're doing that at many different institutions, from Harvard to University of Chicago to Northwestern. Those are two local examples, and with Johns Hopkins as well. But those I think are well known to people, those types of collaborations, but they're very, very important because that is, I think, the germination of where a lot of ideas come from, whether or not they sort of come to AbbVie or to some other organization or whether they go to another place. But anyway, I just wanted to offer a little perspective on the continuum here. I'm happy to be here. I look forward to discussing this with the panel and with the rest of the group there. So thank you for the opportunity.
Oh, and we thank you. That's very, very exciting. And so I must say, as an academician, it's very exciting that this two-way street is really being taken seriously. It's marvelous, which is a wonderful way to introduce Diana Gallagher, who, as I said, is head of external innovation at Biogen, comes with a remarkable history of work of her own, and then through a series of interests finally to Biogen, where she is going to tell us some things about how on earth we should pull this off. Thank you, Daniel.
Thank you, Anne, for having me on the panel, and thanks to the Galien Foundation for the invitation. It's great hearing each of the panelists sort of discuss the complexity, and I would just sort of maybe take it back and say, unquestionably, for us to advance neuroscience, this is a team sport. So there is no ability for us to increase the probability of success and make the gains that we're going to need to make so that we don't see another exodus of folks out of neuroscience if we don't find different ways of working together. And so, we sort of work on that at Biogen in many different ways, and I'll maybe try to think of a few practical examples. So if we think about, some people might think, oh, you have a certain monogenic disorder, and if you have the therapeutic agent, you're well on your way. So for example, we might think of Angelman's, which is a neurodevelopmental disorder in children. We have a wonderful collaboration with Ionis Therapeutics. We understand potentially we could intervene as we have with our Spinraza molecule. And so people think, well, you're quite far away, but all the ways in which even we've had to create collaborations to kind of think about that preclinical development plan and moving into the clinical development plan has been in collaboration with academicians as well as patient advocacy groups as well. So for example, we know we have to understand preclinically we want to hit the target. Well, what's a good way for us to know? We don't understand you could say knock it down, or manipulate it as hard as you can, see how it works. Well, what's a biomarker that we can be using? Well, we found that in the mouse models, the delta rhythmicity as we manipulated the biology would be affected. And so then we looked and we found a cohort of patient samples. This is with a clinical group that actually had a whole set of EEGs, in patients. And we were trying to create the relationship to figure out to help us sort of see how much we want to manipulate the target, how we can pick the dose. And so that's just one element of sort of dose selection and rationale for dosing as we move to the clinic and a biomarker we can use. But that is a target engagement. Then we need to think about the clinical, biomarkers. And so in collaboration, a sort of multifaceted collaboration, we worked on a natural history study, because these children, you're thinking about, people understand that they have seizure. People understand that they have intellectual disability. They have very disrupted sleep. The acquisition of language is, profoundly, altered in these children. But that's not like an FDA approved, that's a collection, of phenomenon. How will we create, whether it's a clinical global impression score, like what is the endpoint, if we do hit the target, that we can demonstrate, efficacy. And so that is in working with the Angelman Syndrome Foundation and FAST, really. And then we did a collaboration with Roche because we said we need to be working together, so we're measuring the same things and can convince the agency we're doing this, in a coherent fashion, because that's what's good for patients and families. So that's just one example. You think, well, this is a pretty straightforward, the target, you understand what you're trying to do. But it really requires, pulling many, many pieces together in collaboration with academics and foundations. And then, another might be sort of in psychiatry, which is an area that I think I'm very excited Biogen, has entered. We have our AMPA-PAM for cognitive impairment associated with schizophrenia. So huge, area of unmet need because although the therapeutics aren't great for positive and negative symptoms in schizophrenia, the cognitive impairment, which often presents in the sort of late, early adolescence, it's very prodromal, and all the patients with schizophrenia suffer from this cognitive impairment. And then we also have the wonderful collaboration with SAGE, is picking up on the thread of postpartum depression and other mood disorders. But when we think about novel targets in psychiatry, again, that is absolutely going to be something where we have to work in collaboration. For us to nominate a new target, people want us to sort of work in collaboration. What's the human target validation, right? And there we have the opportunity to sort of work with, perhaps we can go to the UK Biobank, perhaps we can go to FinGen. We can look at cohorts there. We can then validate it, go over across the street, luckily, and go to the Stanley Center and see, can we do a separate cohort in those? Because really understanding how much do we think we understand about this target. And then we also need to connect, I think in neuroscience, one of the biggest gaps is data. Yeah. There's not a lot of great data. So if we can work with sort of clinicians and healthcare systems, who have electronic medical records, and then maybe we can do collaborations that could be prospective, where we identify the patients. If they have well-curated clinical history data, that's fantastic. Maybe we can prospectively also layer on genomics and then collect information about them, including digital, as was mentioned. That's going to create this sort of virtuous cycle of the phenotypes. And so, those are sort of just two examples. And to pick up on the thread of gene therapy, as we think about CNS gene therapy at Biogen, we have an accelerator that we built in-house, but most of the work has been layering on, collaborations either with startups or recent spin-outs, as we think about, well, how do we get the, different capsids that we want? But also working with neurosurgeons to say, well, right now we don't have a capsid that has the tropism to get, exactly where we need to. What would intraparenchymal, administration look like? What therapies lend themselves to that? What, or if we're going after CNS disorders. So working, with just a host of neurosurgeons as well as other sort of academic advisors to help us think about the sort of capsid to route of administration to endpoint. So, it's something that I think, we will only be successful, if we sort of continue to pull together in this very challenging therapeutic sort of area. And I know that in academics, collaborations at Biogen have always been at the sort of front and center and heart of what we do. And so we'll definitely continue to do that. Thanks, Anne.
Oh, thank you so much. It's very exciting. Whole thing's exciting. So we promised to give a little time toward the end for comments from the audience, and I hope that we can find someone who would want to make a comment. Or are you all? You're not already tired. It's not even 10 o'clock.
Here's one.
Oh, good.
Yeah,
Thank you.
I'll do one. Hi, Maria Luisa Pineda, Visogenix. I wanted to ask about one of the things you guys were talking about, collaborations and data and the necessity for CNS disorders. Then what are your thoughts on the type of, collection or collaborations in order to get that data for CNS disorders? Because that's the issue, right? Every single institution and academic PI is so proud of what they've done, but sharing that data has been extremely difficult. And getting that data out of pharmaceutical companies to share, and even for, trials that have failed, is even harder. So how are we going to get to, for, companies like mine and others, we're putting together, for instance, a consortium so we can federate all these data, genomic, clinical, but it takes every single one of us to get that done. So how are we going to get that to where we need to?
I think Kelsey.
Yeah, I'll start, but I'm sure others have other comments. I mean, one of the things that makes me hopeful is that, in particular, in CNS diseases where one needs a large data set to understand a phenotype, a genotype, that pushes individuals or institutions, academic institutions, to collaborate. So one of the big advantages at UCLA is that we're part of the University of California system. So all of the health systems federated all of their electronic health records. And then we did that with the other the other health institutions in Los Angeles. And the goal was that everybody realized that they needed that very large amount of data to be able to gain insights, make discoveries. So I think that there's that balance between the competition between institutions, which is always going to exist, and between investigators, and then the fruits that one realizes one can achieve by federating information. I would say it's one of the things, again, that drew me to the Simons Foundation, is that funding is provided with the proviso that data be shared. And I certainly have seen that in many, in the NIH and other organizations where I think, if you think about even open publication, the whole movement is towards an understanding that if we really are mission driven in trying to understand, in this case, CNS diseases, we have to collaborate.
Yeah, when I think about this, I think about the possible role or the need for neutral and credible central parties, coordinating parties to do this. I think if you're going to, if either a PI or lab or a pharmaceutical company are going to take whatever competitive risk or other perceived risks or undertake cost or what have you of sharing some of that data, they need to believe there's like sort of a high likelihood that it will actually make a difference, that it will actually be put to good use, and that's the credibility part. And then the neutral part, of course, is that it won't be biased toward one direction or a set of parties versus another. And those, from time to time, there are attempts to build consortia and the like that can do that, but I don't think anyone thinks the full potential of that is anywhere near realized. I would, one thing I wonder, and it might seem far-fetched to some, but I was struck, Diana, we got all the way to you in these introduction remarks before anybody mentioned patient advocacy groups and the importance of those, and I wish I'd brought that up sooner, because they're tremendously promising entities, and I think in the years ahead we might see some of them that have been able to grow and mostly great reputations for neutrality and for impact and for benefit as possible connectors between academia, smaller companies, larger companies, and possibly conveners for purposes of data sharing.
I think it's a really interesting point as people begin to reevaluate, that their data, it's their data, right? The sort of relationship with data will, I think, continue to evolve. And and I do think advocacy groups, and there are already some folks sort of beginning to sort of pull this through to say, for the greater good, like how you use our data, even the natural history, which is so important, even without the intervention, just understanding, how the disease progresses so that we can see the inflection point, it's so crucial. But if every single company is developing, doing their own natural history, it just divides, it divides us. So I think patients and families, and no greater, advocacy groups, especially for rare, severe pediatric, they are like leading the charge, I think, to say, like, we will pull the data because the sense of these are our kids, and the sense of urgency is so high.
I think it's a real stumbling block now, and it has to do with human nature, in the end, and we have to somehow get past that. And I don't know what you all think, but I would think a new effort to do that with pointedly chosen companies, industries, academicians who are willing to do this. This is already starting in the academic world. But what then usually happens is that 10 labs will join, and then the guys outside those 10 labs, of course, they don't know what's going on. So we have to penetrate that. And I think one of the great things that could come from just this sort of conversation as we're having in this panel is that there be a movement toward that. And I think it would be wonderful to have the Galien behind that, because after all, we are out for the human good, even though we have our own. Hello.
How are you?
Let me stop talking because we do want to hear you.
No, obviously we need a global space station.
Yeah.
We need an international space station where we have replicable data across the world. We need to join in clinical research with China and Australia and Europe. And we need to do this politically. We need to have an international, like you said, urgency to this, because this is the biggest global impact on life expectancy and morbidity. And we know Alzheimer's might kick down the door, but alcoholism's right behind, because you can't do a lot of the basic research if you can't have the tools in place that are also helpful for us to do other CNS illnesses. But the advocates often get in our way, and I'm one of them, because we're all siloed by our diagnosis. And we have failed to be given the tools by all of you to show us that what I want for depression is actually very good for neurodegenerative disorders. That affective disorders and neurodegenerative disorders all share this heterogeneity and have this systems analysis approach that is leaving us all siloed. So we all advocate Michael J. Fox, Alzheimer's, addiction, and we're all at cross-competing interests, because no one from the neuroscience community has laid it all out for us and said, you guys are doing it wrong. So I'm just putting that out there, because there's a lot of people in this room who could help inform a lot of the science in these advocacy groups.
I think a fantastic goal. And we have to, as I said, we have to kind of fight our own human nature and get real about this, And even in the COVID pandemic, it's been difficult, which shows you the extent of the problem. But why don't we try, for heaven's sakes, you know? With which we're exactly at the end of our time. It's been wonderful to share with you panelists and to share with you in the audience. I wish we had a lot more time to hear from you. Thank you very, very much.